To be here this morning talking to all of you and to have the opportunity to represent the Rheumatoid Arthritis Challenge. This is a huge group of people work together to make this happen, and so I've listed them here, including our funders and of course the participants as well. All together made this challenge what it was. Okay, so the Rheumatoid Arthritis Challenge is focused on trying to predict treatment response to anti-TNF therapy in individuals with rheumatoid arthritis. So this is an autoimmune disease that leads to inflammation of the joints. It can be both quite painful and quite deleterious to the health, as it sometimes affects not just the joints, but other internal organs. There are 19 therapies on the market today for rheumatoid arthritis, but nearly half of those are actually anti-TNF therapies. And what we find actually is that about 30% of patients actually completely fail to respond to these therapies and are left with fewer alternatives for treatment. So it would be very useful within the clinic to be able to actually identify these individuals before going through the lengthy, sometimes painful and occasionally toxic treatment with anti-TNF. It's also really important as the pharmaceutical industry thinks about what more they can do for these patients to be able to define the population of individuals who are not going to respond to current standards of care, so you can start to enroll those individuals in clinical trials and try to understand what may be beneficial for those individuals. So that's the biological or medical framework for bringing this question to this audience. This was a genetic challenge using genotypes, genetic variants as the features in predictive modeling. So why did we pick that? Well, there was a study that came out last year looking at genetic associations within rheumatoid arthritis. And what they found was that while there was no one individual loci which really seemed to be bearing a huge part of the genetic signal for rheumatoid arthritis, in fact, heritability estimates indicated that about 20% of the variation in the response was in fact attributed to genetic causes. And there was some variation in that across the various anti-TNF drugs that were looked at, although there does seem to be some evidence for most of these. So there's two major kinds of anti-TNF therapies. There are small molecule inhibitors, and then there are antibodies. And so we certainly see that there's evidence of heritability to response to the antibodies, and given this sample size, we can't say as much about the small molecules, but overall, we see that there is heritability here, or there's estimated heritability here. And so given this information and given the importance of understanding treatment response in rheumatoid arthritis, we picked this as a sort of poster child for the field. So there are many treatments out there for which response is non-standard, for which there's great variation in response. And there's many of them for which people in industry and academia are currently looking to find predictive models to help move treatment development and patient care forward. And so if we can take a look at whether estimated heritability actually translates into predictability with models within rheumatoid arthritis, then one can expand such approaches to look across a whole host of other diseases and treatments. So that's really the rationale. Given the sizable estimated heritability here, is it actually possible to use genetic features to predict treatment response? We know this is a polygenic approach, so there's no individual loci, which is really contributing a huge portion of this signal. We know that there are population subtypes, so it's not true that what is the features that may be used to predict in one person will uniformly be true across everybody. And so given this information and the talent within this room here and the greater DREAM community, can we can we say whether genetic heritability in fact foretells genetic predictability? So the format of this challenge was something that I think is familiar to everybody in this room today. We started actually with this group of patients that I just showed you the genetic association analyses on, so 2,700 patients, all of whom are rheumatoid arthritis patients who were treated with anti-TNF therapy. We also generated some additional genomic data that could be used as ancillary data to try to help get a better sense of what may be going on with this disease. And this was RNA-seq data coming from about 60 individuals, rheumatoid arthritis patients who were part of the Arthritis Internet Registry. And so we looked at RNA-seq in blood samples from individuals with severe disease and very mild disease, with the hope that that may help us understand the disease a little better and use that as a biological prior in some of these analyses. So of course, participants were invited to take this data and start to, as their training data, to start to build models, submit models to the challenge. We had a leaderboard, and of course also a completely separate test data set. And so this data set was generated specifically for use in this challenge, and it is a similar set of genotype data, whole genome genotype data, on a second set of RA patients who were treated with anti-TNF. This is from the Corona study. There are approximately 1,000 patients there. Of course, then we score the models and assess what we think is really going on here. This, of course, is supposed to result in a publication, as any DREAM challenge is. And so we did work in a close collaboration with Nature Genetics to assess the ability of this process to indeed improve their confidence in the results. Gustavo mentioned this earlier, this idea of challenge-assisted peer review. Okay, so what does the data actually look like? So in both the discovery data set and the Corona test data set, we had genotypes, common genetic variation marked by about 2.3 million SNPs. There were just a few clinical traits, things that we know are important in predicting both disease and response. And then, of course, we had our response categorical variable. And a part of this was held out from the discovery data set to use for the leaderboard analyses. And then, of course, this was completely held out from the test data so that we could score models. As Gustavo mentioned, this challenge was divided into 2 phases. This was the first time we did this, and so in a sense, it's an experiment in running challenges as much as it is an experiment in rheumatoid arthritis. We had our traditional challenge team phase in which independent teams were able to compete to see who had the best predictive modeling approach. And then we brought people together in a community phase to see whether we could work across teams to further our understanding based on what came out of the challenge. And the challenge was designed into 2 sub-challenges in which we approached this question in 2 slightly different ways. So for sub-challenge 1, we were looking to predict treatment response as measured by change in disease activity score. This is a typical clinical marker of the severity of rheumatoid arthritis, the DAS-28. And it's actually a conglomerate measure which includes both subjective and quantitative traits. And then the second sub-challenge was to actually explicitly identify poor responders. And the rationale, the rationale here was that although predictive modeling is traditionally going to try to look across the spectrum of individuals, what the clinicians really want to know is, all right, who is it that you can tell me with a high degree of accuracy is going to have absolutely no response to this drug? Those are the people I want to target to do something else with. Those are the people who I'd like to keep from having to go through this treatment. The rest of the individuals, if you even think they might have a slight chance of responding, I'm going to give them the therapy and see how they do. I'm certainly not going to withhold it from them. So I'm going to need a high degree of confidence that you've identified somebody who won't respond to the therapy before I will withhold that therapy from them. Here are the team phase results. So altogether, we had 32 teams submitting across these two challenges. And what we found was that, indeed, there was signal. So we were able to predict deltaDAS as a continuous variable with a correlation of about 0.4. And then there was some signal to predict the categorical response as well. And the best methods, the top teams, the Guan lab had the best method for predicting deltaDAS. And there was a tie for the second sub-challenge between the Guan lab and Team SBI. And you will be hearing from both of these teams later in this session. Both of them used an interesting approach to get at this problem. We then moved on to a community phase. And at this point, we said, okay, well, we've seen what you can each do individually. Why don't we all come together and see if we can work to assess what the genetic contribution here is? Because actually, what I'm showing you here is that we can predict response, but what we don't know actually here is definitively whether the genetic information added anything to these predictions above the clinical information that was provided. So this is something that was very important to us to understand. So why don't we come together and see if we can formally ask that question? And of course, we want to see whether when we all work together, it's possible to improve on the predictions that we've already seen. And so we invited the top 8 teams from the team phase to come together and join us in this new collaborative experiment. And I'm pleased to say that 6 of these teams are represented here in this room today. So thank you all for coming, and we look forward to meeting you in person. And so we divided this into 2 parts. First, the teams were asked actually to present their approach and their methodology on a webinar, and this was moderated by an expert in the field, Trey Eidecker. And so the teams got some feedback from Trey, and they got some feedback from each other, and then they were asked to break into groups. And so the 8 teams split into 3 groups, and from there they started to share information about what their processes had been like. One thing that seemed apparent in this challenge was that feature selection was possibly the most important, definitely a very important part of this approach. And so immediately the teams started sharing their approaches for feature selection within this. We gave the team several months to work here, and in the meantime, we had regular webinars. We introduced them to biological experts. We introduced them to the geneticists who had done those original analyses. We gave them what we could in terms of a little bit more information about the disease in the field, which we hoped would help with feature selection. And then we brought everybody back together, and we said, all right, let's all come together in collaboration and decide on an analytical strategy to answer the questions at hand. And the questions that we came up with were as following. So, do genetic features improve prediction relative to clinical models? What is the contribution of feature selection versus the actual modeling algorithm that's being developed on performance? Do we see that genotypes improve prediction when we use biological priors to select them rather than random selection? We certainly hope that's the case. And then an ancillary question, which is, would a supervised ensemble approach improve on individual predictions? And so what I'm showing you here are the results of the collaborative phase, specifically for Subchallenge 1. And so there's a lot of information in this slide, but let's suffice to say that we're looking across each of the teams that submitted. And you can see in blue is indicated their clinical only model. So the predictive model they submitted, which had no genetic information in it at all. And in red is the model they submitted in which they included genetics as they wished. So participants selected genetics. And so we can see that in this case now the best team is in fact Team Outliers, who I believe is represented here today. But we really see no difference between the predictive performance of their clinical and their genetic models. So it does look like within this data, the vast majority, if not all of the information, is coming from the clinical variables themselves. And the genetics really aren't contributing a lot to prediction of deltaDAS. We can look across the various formats. We asked each team to submit predictive modeling, and again, there's a lot of information here. I won't go through it all, but suffice to say that this first bar represents all of the clinical models that were submitted by individuals, and the second bar represents their models containing genetics. And we asked them to provide their own best guess at what genetics should be involved, and then 5 different genetic sets that we had selected that were of varying size, but all of which contained biological information in the feature selection. And so what we see here is indeed, across all of the teams, the clinical models are doing best. They're doing better than the participant selected genetic models. And then as we add additional SNPs, as we increase the feature size of the genetics across this, in fact, the performance gets somewhat worse. In this case, again, we did perform an ensemble analysis using these results from these teams. Actually, it's the results from the team phase. And that did not do as well as the individual predictions within this first sub-challenge. So if we look at the second sub-challenge, which remember is the clinically relevant one, which is predicting non-response, we see a different story. So in this case, again, there's not a huge sweeping difference between the genetics models, which are, again, are the red ones, and the clinical models, which are the blue ones. But in fact, in this case, we see that the best model, in fact, we do see significant improvement, significant although small improvement of including genetics in predicting non-responders. So this is encouraging. This is something we're following up on a little bit more. And in this case, when we take a look across the various types of genetic features we asked people to build their models on, we see less of a pattern. So it's less true that, in this case, the clinical and the participant models are pretty similar, although in fact the participant models are doing better, and we see less of a trend related to the size of the genetic features used in the models. In this case, however, the ensemble analysis, and that was done by Gaurav Pandey, who I believe couldn't be with us here, but we could certainly get anybody interested in contact with him if you'd like to talk a little bit more about supervised ensembling models. His model, in fact, did better than any of the individual models here. So in conclusion, we found that what I didn't mention was that both of the teams that won in the team phase use a Gaussian process regression approach. And so we'll hear about that from both of these teams this morning. And so this seems to be best in terms of this kind of question. We found that indeed SNP selection did seem to be more important than the selection of the modeling algorithm in most of these cases, and that genetic information does improve prediction for non-responders over use of clinical information, although it didn't do as well when we were looking at continuous response across the whole spectrum of responses. And so we're currently assessing the ability of these models to predict response relative to what a clinician may actually need within the clinic. And we're doing that by working with a set of clinicians to themselves assess what they would predict for these individuals, if they can predict non-responders, and whether these algorithms in fact do any better than that. And this is also opening a dialogue with clinicians to say, well, what is it, given what we've been able to find with genetic information, we think that there may be other modalities we can include in this question moving forward that may be useful, and where do you think lies the best promise for signal moving forward? So with that, I would like to turn it over to, actually I'd like to turn it over to Stephen to present the awards to the individuals who did win this challenge, had the best independent team phases, and then we'll hear from each of them.